Well, thank you very much for the very nice introduction, Ellie. That was very kind. So very excited to be here and to talk about what we've been doing in terms of creating a pipeline for comprehensive molecular profiling for CNS tumors. So I am at the German Cancer Research Center in the Neuropathology department and at the University Hospital of Heidelberg. These are my conflicts of interest and disclaimers. So as most of you who probably work or also do not work in CNS tumors might know that the new or the current WHO classification 2021 is the most molecularly inclined of to date of all the classifications that we've had. Now, what this really means is that in order to make an integrated CNS tumor diagnosis that is compatible with WHO, one needs to report a wide variety of alterations, which you can see quite nicely over here. So, of course, you have the normal, well, not quite normal, but the usual things like mutations, copy numbers, etc. But you have methylation classification that Ellie spoke about, which was pioneered in Heidelberg, which was way before my time, so I'm not tooting my own horn here. But what we need for this, right? I mean, if you want to make this sort of diagnosis, you need to run a wide variety of assays, which you can see quite nicely here from this review from Beretta et al. 2 years ago, and where they really tried to pinpoint what are the different assays one needs to run, in what sequence, in what quantity, really, to make such a comprehensive diagnosis. And of course, this is labor intensive, as you can imagine, but there's a bigger problem. There's the initial setup for some sort of a facility that can run this is over €1 million or dollars, depending on your jurisdiction. And the bigger problem in centers that even have everything, right? So at Heidelberg, we do have this whole setup, but the turnaround time, even for us, is maybe 3 weeks, could be more, especially if you have send-ins, it's probably much longer. And this is probably around like 2020 or 21 when we started thinking of, we need to have a solution for this, because we kept increasingly getting questions from other institutes like, well, what are we supposed to do to be compliant with WHO in order to make these diagnoses? And so that's when we started looking at Nanopore as a solution. So I'm not going to go into what Nanopore is. I think you've heard a lot from Ellie, and we're probably already familiar with how this works. But the main sort of selling point for us was, of course, these small devices, but also the ability to detect methylation, right? And already, I think, in 2020, there were a few papers that had come out about using the DKFZ dataset and classifier with Nanopore sequencing. But these came with some limitations because they were low coverage, whole genome. You could not detect SNVs. You could not get very good CNV resolution to the extent that we've been getting with arrays or NGS currently. And because it was very non-uniform CPG site coverage, we could not apply all the state-of-the-art methylation classifiers that we've been developing directly on them. And that's when adaptive sampling really came into play. I think in 2021, when Matt Luce had a paper in introducing ReadFish, which is this really interesting tool to enable adaptive sampling. And what is this adaptive sampling, right? Essentially, you give your sequencer a text file saying, okay, I need to know, IDH, TP53, BRAF, etc. And what happens is, when the DNA is going through the pore, so in all the pores on your flow cell, within 100 or 150 base pairs, the system has already read it, aligned it, and made a decision whether this is in your target or not. And if it is in your target region, then it continues sequencing. And if it is not in your target region, it rejects it. So what you're really doing here is you're doing targeted enrichment for genes without primers or without pre-planning months in advance what is it that you really want to sequence. And the way we do it in Heidelberg now is, of course, we start with a standard panel, but sometimes midway through, a few hours later, we're like, I know what tumor type this probably is. We just sequence, 5 genes now, because those are the ones we really care about. So it's very flexible. So we took this and we developed 2 methods. So the first one is, of course, the more important one, which is called Rapid CNS Square, which is our pipeline for real-time nanopore-based profiling of CNS tumors. And you can see the schematic here. So what we can do, and what we do, is we get tissue directly from surgery, and already intraoperatively, while the surgeon is still sort of, working on the tumor, we start sequencing. And within 30 minutes of sequencing, we can already give a broad methylation class and broad CNVs. And then you continue sequencing for about 24 hours, and at the end of it, we have this comprehensive profile which says, which are the clinically relevant mutations, CNVs, structural variants, MGMT promoter, etc. And to also complement this with the latest, most up-to-date methylation classification, we developed MNP Flex, which is a classifier that classifies 184 subclasses of CNS tumors. And we performed a very large validation study, which was also my PhD thesis. So I'm going to start off with the intraoperative results. So first, what we did was, of course, to assess feasibility of this method at all. We did a simulated run. So we took samples we had already sequenced before, and you can see the results here. So the x-axis here is the time in minutes, and the positive y-axis are the correct predictions at that particular time point, and the negative y-axis is the incorrect predictions at that time point. And you can see, okay, within 30 minutes or so, right, you have a lot of samples that are correctly predicted. But what I would like to draw your attention to, which is very important for us, and I imagine also for you as pathologists or working in diagnostics, is that the scores here, so the green score boxes, are basically the scores for the correct predictions, and the orange for the wrong predictions at that time point. What you can see is that the green boxes are always much higher, so the scores are always higher for the concordant ones, and the discordant ones are always very low. So we never made a wrong prediction with a high confidence. And this was also then pretty much reproduced when we did real runs in Heidelberg and in Nottingham in tandem in two independent institutes. We found the same thing, that within 30 minutes we could resolve most cases, and the scores again were pretty well separated. So we were not really giving wrong information confidently to the surgeons. And this is what it basically looks like. So this is one of the cases from Heidelberg, which is a glioblastoma, and ependymoma from Nottingham, which was already classified correctly within 5 minutes, and then kept getting a consistent score up to 1 hour of sequencing. But as you, of course, saw from the previous results as well, right? Like, there were a few which we just could not resolve with methylation. Like, they never became correct. This is one of these cases we had in Heidelberg, which, as you can see over here, it kept giving us all sorts of weird results with very low scores. We would have never taken it. But what we also got was, of course, these copy number plots. These are for every 5 minutes, from 5 minutes to 20 minutes onwards. And you could very clearly, in these plots, already from 5 minutes and consistently up to an hour, see a 7 gain and a 10 loss. And we knew from the clinical data that we had for the sample that it probably was a glioma, and with a 710, we could say it was a glioblastoma. And like I said, of course, we continue sequencing, so we just don't do it intraoperatively. We want to have a full picture for the diagnosis. And so we first looked at how does this perform for SNVs, which was something really important for a lot of pathognomonic alterations. So here you can see on the y-axis is the percentage of SNVs that are recovered from NGS, which was pretty high. I mean, the tail that you can see are all samples that are from the old flow cells, which, of course, admittedly had a much higher error rate. And we wanted to see then, what is the on-target coverage that we need? Because, of course, there are different numbers that are floating around all the time. What we found was that about 15 to 20x on target is already enough to recover about 90% of the variants that are reported by NGS. Next, we looked at the CNVs. So, of course, the intraoperative ones were more broad. But now we want to look really at focal alterations, like CDKN2AB deletion and more small alterations that we have. And to the left, you can see for the same sample, the methylation array profile, and to the right, the rapid CNS square one. And again, arm level are pretty well reproduced, but the focal ones as well were very nicely recaptured in rapid CNS. And I think this is one of the strengths of this method, that the CNV profiles are very concordant. And of course, then we looked at MGMT promoter methylation, which is very relevant for temozolomide resistance. And what is very useful, I mean, well, not at the moment, right, but I think in the future it will be very useful, is that rapid CNS, it covers the entire MGMT promoter region. So this blue background, right, the highlighted part is the MGMT promoter region, and the methylation profiles for 4 samples, and without me really telling you, you know which ones are methylated, which ones are unmethylated, because it's just so well separated here. And like I said, we right now only use a few probes on the arrays, on pyrosequencing, etc., but this could be a resource that really helps us to understand temozolomide resistance probably better once we have enough data generated. And just looking at the concordance, we found that more than 90% samples were correct as compared to the EPIC array prediction, which is currently the gold standard. Next, of course, I mean, long reads, right? So we were looking at structural variants to see if that is much better, or at least the same as what we get with NGS. And we found that in 7 pediatric astrocytoma cases where we had RNA sequencing data as well, matched with NGS, we found all of the BRAF-KIA fusions, which are, again, pathognomonic and targetable as well, that we could find them in the Nanopore data as well, pretty reliably. But we also went a bit further, right? We're like, okay, I mean, these are things we knew from NGS. What can Nanopore give us that's more? And we looked at EGFR, and you, of course, probably are familiar with the EGFR variant 3 in glioblastomas. And we found one, actually, in a glioblastoma sample, which was not detected by NGS. So this was something we did not expect. We also went back and checked. We did not see it. And it was subclonal. So you can see the IGV screenshot here. And there's the exon 1 and the exon 7. So you have the 2 to 6 deletion. It was only in 18 reads out of 100 reads that covered this region, and this is basically the same read, and there are many of them. So it was a very confident deletion. But again, like I said, NGS did not report this, so it had to be subclonal, probably acquired after the amplification. So essentially basically trying to say that, there's so much more we can find out from this data than just, trying to find what we already had with NGS. So it's not just a replacement, but could be an improvement even in some regards. So what we did was, because we wanted to see how does this exactly fit into things, we took the diagnosis that we had from conventional analysis, so with the array, NGS, histology, etc., and we gave the Nanopore reports to the pathologists, and we tried to see if they would come up with a similar diagnosis that was already recorded for the conventional methods. And so essentially we did this for 301 samples from 2 institutes, so from Heidelberg and from Nottingham. And you can see the very complicated schemata here. But essentially what I'm trying to show you is that if the methylation class was correct, of course you finally most likely got the correct diagnosis. But in the cases where it was either compatible or a mismatch of methylation class, which can of course happen, because rapid CNS gives you mutations, it gives you CNVs, fusions, etc., we could resolve these cases to finally get a concordant integrated diagnosis. And what was really important in this study was that we had a less than 2% misleading rate. And I mean, we say misleading, we don't say discordant, because we had all this data at the same time in the Nanopore reports, in these 5 or 6 cases that were misleading, it never presented a very convincing picture of a wrong diagnosis. It was always conflicting because maybe the SNVs were not quite concordant with what you would think the CNV should have been like, or the methylation. So it always led to us, not us, sorry, the pathologists, asking for more tests, maybe targeted sequencing, more immunohistochemistry, more clinical data, etc. So it would add a lot of procedural safety that we usually don't have when you have consecutive tests that are done. And then, like I said, of course, we developed a classifier, which we call MNP Flex, which started off, honestly, as a classifier for rapid CNS data. But then we got so much data from different people across the world that we tried to make it more platform agnostic, so every sort of methylation data could be used for it. And here you can see essentially everything we validated it on. So it's about 5 sequencing technologies, or 4 sequencing and the methylation array. And we validated this over 78,000 samples with a subclass level accuracy of 99%. Right, and how does this look like in practice? This is basically a case from Heidelberg, recently, I think. So what essentially we did was, of course, we start sequencing. In 30 minutes, we could say it's a meningioma with a 22q loss of heterozygosity. And after 24 hours, when we had MNP Flex, we had all the mutations, etc., reported, we could say it's a benign meningioma with these particular SNVs, CNVs, and unmethylated MGMT, which, I mean, it should be, so that's good. And why does this matter? Because this pipeline then enables intraoperative characterization, which is very useful for guiding surgery and for guiding extent of resection, which is very important, especially in pediatric tumors. It reports the whole spectrum of the WHO-relevant alterations, which is then sort of giving a one-stop solution to this whole complicated web of assays that you need to run otherwise. It enables granular state-of-the-art methylation classification, so with MNP Flex, not just with Nanopore, of course, but with other platforms as well. So you're not restricted if you want to do methylation classification. And because it's small devices, you don't need to batch, and it's also easily targetable regions, it's very scalable for use in under-researched regions, which is very much going towards our shared aim of making molecular diagnostics more globally accessible. And with this, I'm at the end of my very long talk. But with that, I'd like to really thank everyone involved in the study. It was a lot of people, very huge effort, especially Felix Zahm, who was my supervisor, and Andreas von Daimling, who's the head of our department, for really letting me experiment a lot with these very tricky, very experimental solutions that I came up with. And thank you for listening. I'm happy to take questions.